in dwarf and normal Wisconsin fast plants. So just quickly, gibberellin is a growth hormone in plants. It's a protein. So my abstract is the type of plant that I'm using is a Wisconsin fast plant, and I'm using a dwarf variety and a tall variety, which is just the normal heighted variety. And basically what I'm doing is I'm growing them in the same environment, and then I test their protein levels and I test their specific gibberellin levels. Then I put them each in different environmental conditions based on light, dark, and rich soil and dirt. And you can see over there the 4 different combinations, and I'm going to do 2 of each sample size, so I have a larger sample size. And then once after those plants have been grown in each of the different environments, I will again test their protein levels and test their gibberellin levels and compare them to see if there are any differences in between the original and the after the different conditions, and per condition see any differences. And to test the overall protein amounts in the plant, I'm using the GOT protein kit, which uses the Bradford method. And for my testing the gibberellin, I'm using a gibberellic acid ELISA kit. So my hypothesis, I have sort of 2 different hypotheses based on the tall and the dwarf variety. So for the overall protein levels, it's not a quantitative amount because I don't really have any idea what the spectrum is. But in comparison, I think I conjecture that the plant that is exposed to light and has enriched soil will have the highest protein levels, and then the next highest will be in the darkness with the enriched soil, and then the light and the dirt, and the dark in the dirt. And the same goes for the comparative gibberellin levels, and this is for the tall plant. So for the dwarf variety, I predict that overall they're going to have similar protein amounts, smaller, but they're going to have a lot less gibberellin because that's the gene that I expect to cause the dwarf variety. And the order is the same for the overall protein: light exposure and enriched soil, dark and enriched soil, light and dirt, and dark and dirt. But for gibberellin, I expect no detectable change in the gibberellin levels based on the different conditions that each plant is put in. So GOT protein is the kit that I'm using. It uses the Bradford method to determine the protein concentration. So the procedure is that I have to get a sample tissue from each sort of plant, from the same place in each plant, because could be that the stem might have more protein than a leaf, and so I want to make sure that that's consistent throughout my experiment. And then you mix it with a PBS buffer, and you add this dye called Kumasi Blue, G250 dye, and basically what this dye does is it bonds to proteins. So the more proteins, the more binding there is. And then I put the sample in a cuvette, I put it in a spectrometer, and then I look at what wavelengths are absorbed by the solution. And so basically the way it works, like I said, the dye bonds to a protein, and so the more protein it is, the deeper the blue, because there will be more binding. And so basically it uses Beer's law that states when a solute absorbs light of a particular wavelength, the absorbance is directly proportional to the concentration of the solute in the solution. So that's basically how it determines comparatively what has more binding to proteins and less binding. And the more binding there is, the more protein there is. The less binding, less protein. So why am I using the Bradford method? Why is this the best method to use? So there are 3 different ways to quantitatively measure protein amounts. There's the Burette and the Lowry and the Bradford. So with the Burette, it's a chelation and a redox reaction. The Burette isn't as accurate as the Bradford method, and the Lowry has interferences from a lot of common chemicals that are found in the lab. So, that are just in that room right there might affect the results. And so the Bradford is also the most accurate of the 3. So gibberellin, this is a picture of gibberellin's chemical structure. And basically it's a protein hormone, and it helps plants grow by inducing cell growth. So the larger the cells are, the larger the plant will be. And so a gibberellic acid ELISA kit will be used to determine specific gibberellin amounts versus overall protein amounts, because there's more proteins than gibberellin in a plant. So the ELISA procedure is I have to, again, collect the tissue sample, again, from the same places on the plant, and then I purify it by centrifuging. I have to add 100 microliters of each sample into different wells in a plate, and I have a plate sealer that has certain antibodies on it, and basically how it works is how it specifically targets gibberellin is these antibodies on the plate sealer are specific for gibberellin to bind to the detection reagents. And there's 3 different detection reagents, and basically the color will change like it does with the Bradford method, and then you add a stop solution to stop binding. So you can do that sort of at the same time to make sure that your results are consistent. And then you, again, measure the wavelengths of the colors using a spectrometer. And so this is a picture of liquid nitrogen. Liquid nitrogen is what's used to collect your tissue sample. Why am I using ELISA? So this specific gibberellic acid ELISA kit, like I said, the antibodies are specifically target gibberellin, so I won't have data from other proteins. And it avoids data from other proteins in the plant by using a wash buffer and other things that might be in the lab. And then again, it uses a spectrometer to measure wavelengths. So what other research has been done on gibberellin and protein? And so a study was done by Metzger in 1984, so kind of a long time ago, when they were sort of discovering, why is gibberellin important. So there's a plant, Philosophy arbens L, and it grows in cold environments. So he replaced the cold environment with gibberellin to see if it would still grow, and he concluded, because there was equal growth, that gibberellin is a key protein that stimulates plant growth. And then another study was done by Rademacher in 2000, and basically it studies the effects of chemicals on gibberellin growth. So I'm studying environmental effects, he studied chemical effects. And then each of these following chemicals provided growth inhibition for the gibberellin protein. So ammonium compounds with an N-containing heterocycle, structural mimics of 2-oxoglutaric acid and 1617 dihydrogenate 5. So what are the benefits of my experiment? Why is it important to know how environmental conditions affect gibberellin protein? So if my hypothesis is proven incorrect, meaning that environmental effects have no causes on gibberellin, hydroponic growth companies, which is where they grow plants in rooms with no soil, can use fewer light resources and therefore decrease energy usage. And they have to, and for people who, not hydroponic, but people who do use fertilized soil, can use less fertilized soil, and which will end up decreasing carbon dioxide emissions through cow reduction. And so if my hypothesis is proven correct, meaning that environmental conditions do have an effect on protein levels and growth, then researchers will know to continue to research ways to optimize products using the least amount of resources. And I'm excited.
Okay, so you're going to quantify, well not quantify, I guess you're going to look at the relative amounts of proteins, and then you're going to detect, the ELISA kit detects whether the gibberellin is present or not. Does it?
It quantifies it.
Oh, it quantifies it. Okay, I guess that was my question.
Yeah. How? Using the spectrometer. So,
So you mentioned having...
So when you add the detection reagents, based on what the reagent is, it will change color, and it's again relative, binding relativity, and then you use a spectrometer to measure the wavelengths of the color.
So So it's not just detecting presence or absence, it's also detecting quantity?
Yeah.
Okay.
Is the total volume going to be significantly more than 100 microliters?
But
Is the total volume of the sample going to be significantly more than 100 microliters? Because the spectrometer that we have won't detect 100 microliters.
I could probably find a way to sort of make the ratios same. So if I needed 1,000 microliters, I could just multiply my other solutions by 10. So it's the same ratios. Try that out.
So for the dwarf plants, as far as gibberellin levels, you're thinking regardless of what, like how they're grown, the levels are all going to be the same?
Yeah.
So what?
And they're not going to change from environment to environment. From the original environment to the new environment, it won't change, and for each different condition, it will be the same.
Okay, so why does it change for the tall plants and not the dwarf plants?
Okay, so the tall plants, the reason that they're tall, and the dwarf plants, the reason that they're tall, is their genotypes are different. So the tall plants have the genotype for the gene that causes gibberellin production, and since the dwarf plants don't have that gene, which is why they're dwarf plants, there's nothing to change about their gibberellin production.
So basically the dwarf plants have as much gibberellin as they have, and there's no more production and no more expression. That's just the level, or do they have none?
Yeah, or that maybe a different protein has caused their growth.
Okay, got it.
Is that why you're studying the other proteins also?
Yeah.
Because you're doing two tests. You're
Yes.
doing one for, so,
To see if protein levels, to see if protein levels overall change, which I think they will for the dwarf plant.
And then if you see that they don't change, it might be that another protein kicks in, other than gibberellin, if you found that it was a
Right.
present.
Yeah.
Are there any studies to show what other proteins might be involved in height?
I
or
didn't
growth, or-
find any. I think gibberellin is a pretty
Maybe.
consistent
Okay.
protein.